I am a senior scientist here at Biosolutions, and really what I get to do is I work a lot on the development side of the projects here, meaning that if we have a customer request or something that's going to be a custom project, I usually get to develop some of those assays, some of the protein purification. So the way we approach protein purification here at Biosolutions is through a number of different methods. Usually they are lateral methods, so we may have a protein with a certain isoelectric point that we need to separate from proteins with other isoelectric points, and one of the ways that we do that is through ionic exchange chromatography. We use anion and cation exchange chromatography to do those types of separations. But sometimes we may have proteins that have similar isoelectric points, but they may be different sizes, where we can then separate them based on their molecular weights and do gel filtration chromatography. A lot of times we may still have proteins that are similar in PI and similar in molecular weight, so we need a more affinity method, which we have several different types of that type of chromatography here at Biosolutions. We usually test for purity at the end, so we know ahead of time what type of purity we're trying to achieve. Many of our proteins are greater than 90% purity or even greater than 95% purity. A lot of times we use SDS page analysis, which is sodium didecyl sulfate acrylamide gel electrophoresis, to test for the purity. So the way that we do that is we take these proteins of interest and then we resolve them according to their molecular weights. And if we have a pure protein, say it's a 50 kilodalton protein, and we have a band at 50 kilodaltons after we stain the gel and we can't see other proteins, we know that that's our protein of interest. It's free of contaminants. If we see multiple bands on the gel, we know that it needs to undergo another round of purification to increase the purity level. And a lot of times we use another process called densitometry, which is where we scan in these gels and then compare the different bands in each lane to see what percentage is that large protein that we're looking for and what percentage is the other contaminants. And that's how we decide if that protein is pure enough to be sold to our clients.